So thank you very much for inviting me to come here and to speak at this illustrious conference. I'm very pleased to be here, and I thank the OSA student chapter and the OSA for making this possible. And so if you are budding physicists who have an interest in photonics and optics, I do encourage you to go to the website of the Optical Society of America and take a look at what they do. They really are promoting science, young scientists, and also older scientists who have an interest in optics. It's a fantastic organization, and at the moment it celebrates its 100th year, its 100th anniversary, so there are lots of extra activities that are going on. So without further ado, I just start with my talk today, which is entitled Superresolution Microscopy. So what I do is I give you an overview of the principles of how one achieves super resolution, which is classifying those methods of microscopy, optical microscopy, where you can look at things that are much smaller than the wavelengths of light. So I will give you two different technologies. There are several now, but I will only touch on two because we only have 3/4 of an hour. The first technique is called single molecule localization microscopy. And I will talk you through the principles of how this works, and then applications from our own laboratory of this technique, where we look at problems of neurodegeneration, how neurons die in the process of proteins going crazy inside cells. And then the second super resolution method that I will talk about is called structured illumination microscopy, where you project patterns of light onto your sample and generate spatial frequency beats, and this allows you to gain more information about your sample from a microscope than you would otherwise get. And I will show you some examples of that too in biology. And before I do that, I'll give you an introduction just to show you briefly what degrades image quality in microscopes, so we understand these techniques in a little bit more detail. And so I'll particularly go into these concepts of point spread functions and numerical apertures, which will set the scene for the rest of what I talk about. So I should thank my group, whose work I'm partially presenting here today, and our collaborators, who are in touch. And again, I would like to thank the OSA for making this possible and funding this. So here's my last poster advertisement for the OSA. I was asked to do this, and I do this with very happily. So the OSA has done a lot for me personally. I'm a fellow of the Optical Society of America, and throughout my career I've benefited greatly from them. They organize the best conferences, in my view, on optical and optics related topics. So do go and check out their website. All right, so let's start. Let's get into the meat of things and talk about different things. Talk about point spread functions, numerical apertures, image resolution, and all that. So what are we dealing with when we do optical microscopy? If you want to study biology, you want to study living things, living objects, and what better way to do that than using light. If I probe something with light, I can keep it alive if I do it properly. So as long as you have an object where you can interrogate molecules specifically and sensitively using the right colors of light, then you can use optical microscopy and you can look at small things. But of course, the problem is that the microscopes really haven't changed all that much from their first implementations. So this is a picture of the first compound microscope. A compound microscope, and the principle of it hasn't changed. If you buy a modern research microscope from Leica or from Olympus or from Zeiss, the components in it are almost identical to what you see here. You have a condenser lens and a light source. The light source here is a flame, so there's paraffin inside this bowl here. It lights this candle here, and then you have a water-filled bulb here, which acts as a lens. And the light from that, from the flame is then condensed onto the object, which you can't see here. There's a little needle down here which contains the object. And then there's the microscope objective. That's a high power lens. And then there's something in here which today would be called a tube lens. And that tube lens then projects an image onto whatever is behind there, usually a camera. Okay? And so in practice, the microscopy system looks like this today. You have an objective, and you have a tube lens. You have a sample which you place a focal distance away from the objective. And then you have the tube lens here. So in the back focal plane of the objective, you place a tube lens one tube focal length away from that back focal plane of this objective here. And then you have an image plane, which is another focal plane away from the tube lens here. And so any sample point will give you a corresponding image point over there. And of course, you old physicists, you know that if you go from one focal distance to another focal distance, you essentially get the Fourier transformation of the aperture function, whatever is on that side. So we can look at this like that. If I have the object plane here, and let's say I have a sample containing lots of fluorescent molecules. Here's one such molecule, right? This molecule here will now be emitting light into all directions. Some of that light is captured by the objective. And so you can see a point source emits spherical waves. The spherical waves are partially collected by this objective function. They're turned into plane waves because we are just one focal distance away. And so if I take this point here, I can represent it with a delta function, right? It's just a point object. And you can see that this is a Fourier transform here because here you have a plane wave. It's infinite in all directions. What's the Fourier transformation of a delta function? It's an infinite. It has infinite frequencies in it, right? So essentially what you have here is an infinite extent of frequencies, right, in this direction here. So a delta function has a Fourier transformation that's unity over all frequencies. Okay? So here we have that, you see this. And unfortunately, we don't collect all of the light. Not all of the spherical wave is collected by this objective here. It's only collecting over a finite aperture. So this Fourier transform is clipped. Okay? So you can see this here. So we're clipping it with the top hat function. So this top hat now is removing frequencies, spatial frequencies here and spatial frequencies here. So think about this plane here as being the spatial frequency representation of this object point. A point has infinite spatial frequencies in it. And so here we're clipping some of that frequency information, and then from that clipped information we actually we form an image point. So this is again focal distance. And so you see here what we now get is we have a product of a unity function here, infinite spatial frequency extent. We clip it, and then we take the Fourier transformation with this lens. So what we get is now a Fourier transform of this, which is again a delta function. That would have been our original point. But unfortunately now it's convolved with the Fourier transform of this. And that here is called the point spread function. So what you get is a degradation. So by those of you, you're all physicists, or most of you are, so you know that a product in real space becomes a convolution in Fourier space. So this product of this function here with this rectangular function becomes a convolution of the Fourier transform of this function, which is the delta function. That's the original information, times the Fourier transform of this function here, which is called the point spread function. Okay? That's what degrades your image information. So what you have to remember then is if I take a point, I image it through a lens onto an image, then that point will not be a perfect image of that point. It will be spread out. Right? I've degraded it because I'm only sampling a finite amount of the spatial frequencies of the object. And so Abbe, at the turn of the 20th century, was the first to recognize what the resolution limit is, because of course every point is now spread out, and he recognized that there's a finite distance over which I can separate two object points in the image plane, because if each point gives rise to such an image here from the object, then 2 points situated next to each other will become indistinguishable if these point spread functions come too close to one another. All right? So he found out that the distance, the spread in space of this point spread function is given by lambda over 2 times n sine alpha, where sine alpha is the collection angle of my objective lens, right? So the larger the angle of light, the more I capture from each point, the smaller the size of this point spread function, right? And the better the resolution. The best resolution with the best lenses that we get, so this angle here is related to the numerical aperture of the objective lens. That's why in a microscope the sample is always very close to the objective, right? So you can capture as much light as possible, so the angle of light that's captured from every point is as large as possible. The numerical aperture needs to be large. So good microscope objectives may have numerical apertures of 1, even above 1.4 with the best lenses. And so you can see that the maximum resolution is roughly lambda over 2. The wavelength of the light that that point emits divided by 2. So let's suppose I've got green light, like this, so let's say it's 500 nanometers in wavelength, lambda over 2, the point spread function will be roughly 250 nanometers wide. That's the best I can do. It's not surprising, I'm imaging with light waves, right? It's not surprising that I get something that I won't get resolution much better than the wavelength of light. That's what we expect, right? Right. How do we go beyond that? Cells have very small objects, an organelle inside a cell, a whole cell maybe of the size of 10 microns in size, 10 micrometers in size, an organelle inside the cell maybe of the order of 100 nanometers in size. That's smaller, much smaller than the wavelengths of light we're using. How do we have any chance of resolving this stuff? The first technique is very simple to comprehend. It's called single molecule localization microscopy. And in 2014, this technique won Bill Murner from Stanford and Eric Betzig from Janelia Farm the Nobel Prize in Chemistry. And so if you will see, I was say painting molecules with optical pointillism. You know, pointillism is the style of painting where you make little dots, right? And when you go very close to a picture, you see nothing, but when you step back, you see the picture emerge, and you will see why this is appropriate to describe the principle of this technique. So to illustrate how it works, I have this cartoon here, which is done in the computer, so this is not a real image, but assume I have an image containing lots of fluorophores. So there's bright where there are lots of fluorophores and dark where there are no fluorophores. So this image here is produced by lots of molecules emitting light. It has a size of 2 microns. This scale bar here is 2 microns. And so if molecules, there are lots of molecules sitting here, and I'm imaging those molecules with a microscope, this is what I would see in a good microscope, in a standard wide field microscope. So all these fluorophores are emitting. So imagine there's a fluorophore sitting here. In the image plane, it gives me a 250 nanometer spot. The molecule next to it, 250 nanometer spot, but they're all overlapping. All these spots are overlapping, I get image blur. So by switching all the molecules on at once, the point spread functions all wash out with one another and give me image blur. So instead, consider only switching on very few molecules at once. if instead of switching on all molecules at once, I just switch on a sparse subset, in this case 2 molecules, then if I manage to keep the density of fluorophores that are on at one time very low, then the probability of 2 point spread functions to overlap is very small. So there's a molecule here which gives me a point spread function. The width is lambda over 2. This one here is a molecule, gives me an image of with a point spread function lambda over 2, right? But I know that the molecule that gave this image is at the center of this distribution. And I know that the molecule that gave rise to this image is at the center of this distribution. So all I need to do is to fit the point spread function to this photon distribution to get the coordinates of this molecule, right? So I record the point spread function of this molecule, and I record the molecule's location by fitting the point spread function. All I need is the peak of that distribution. So it's somewhere in the center here, I draw a dot. Somewhere in the center here, I draw a dot. And I keep doing this lots and lots of time, take lots of images, and then I can reconstruct information that is much more highly resolved than what you had originally in the image, okay? Because now the image information, it's not a real image in that sense. You have to think about, this is a reconstructed image. So the image information, what you really have is localization information. You've localized all the fluorophores in your image. And what you've done is you have taken, you have separated the information in time, right? By taking lots of images rather than taking all the molecules at once, you can separate out the point spread functions spatially, right? All I need to do is make sure that the density of fluorophores that are on are so low that the point spread function overlap is very small. So the precision that I get is just determined by my ability to fit the point spread function to each image distribution. And that precision is only given by the number of photons that I have per point spread function. So if I have an original point spread function, let's say it's 300 nanometers, the image width is 300 nanometers, how accurately, how precisely can I localize the molecule? It's given by the original width, 300 nanometers, divided by the square root of n, where n is the number of photons per point spread function that I image. So if I have 100 photons, I go from 300 nanometers divided by the square root of 100, which is 300 nanometers divided by 10. So I go down to a localization precision of 30 nanometers. With 10,000 photons per molecule, I get a localization precision of 300 divided by 100, which would be 3 nanometers. Of course, there are other factors that then come in, but routinely we can localize in the laboratory now with typical fluorophores that we use in biology to about 10 nanometers in size. And what that does in practice, so it's shown here. So here's an image that we've taken of a protein called amyloid beta. Amyloid beta causes Alzheimer's disease. And so it's a protein that misfolds. So it doesn't adopt the correct shape, and then it starts getting sticky, and the protein will then start to self-associate into long fibrils, and in the process of this becomes toxic and causes neuronal degeneration, so neurons start dying. And here you see a wide field image of this protein amyloid beta, and so what you see is that here's a scale bar, 500 nanometers. So all these things have these are labeled with a certain dye. These amyloid beta proteins, but you don't see very much because, of course, all the molecules are switched on at once. All the point spread functions overlap, and you get image blur. So now instead of doing this, let's reconstruct with the single molecule localization trick, and then the result, this image turns into this. So you can see a drastic improvement in the in the image resolution. Now we can clearly see this is the same image taken with this technique. This is one variant of the technique that's called D-storm, direct stochastic optical reconstruction microscopy. And so maybe we can turn the lights down a little bit because some of the, is that possible? Just a little bit, so that these images become clearer to see. So, but actually, there are a whole bunch of techniques which are identical in their principle. They're called PALM, STORM, F-PALM, Palmyra, GSDIM. The principle is all the same, right? Fiona is another one. So they're exactly identical. You localize individual molecules from their point spread functions and get super resolved images like this. So you're trading speed for resolution, and then you get images like this. So here you see this process that we are interested in. There are several examples coming, this amyloid formation, so that in the future. So if we have a peptide that forms a protein that comes from the ribosomes, if this peptide somehow doesn't adopt the correct shape in this process inside the cell, it will expose beta sheets. It will adopt a beta sheet structure, and these beta sheet structures can stick to one another if they have hydrophobic surfaces on the outside and form these long fibrils. They're almost crystalline in their appearance, beautifully rigid fibrils, very strongly bonded by hydrogen bonds between them, and these can cause you serious problems. And they're responsible, for example, for diseases such as Parkinson's disease and Alzheimer's disease. And some of the questions we would like to know is, where do these species originate? How do they transport through the brain? Are they becoming infective to other proteins inside the cell? And of course, if you want to study this process inside cells, then you want to do this with optical techniques, right? Because with optical techniques, we can be very selective. By using special molecular labels, we can just target the proteins we're interested in, not the others. And of course, we can look inside the environment where these species are formed. So these are some of the research questions that we would like to address. So the previous picture, you could have done that with atomic force microscopy, right? It's just a cover slip with protein on there, and you watch these molecules grow. So what's the purpose of using optics? But the next thing you can't do with other techniques, and that's looking inside cells. So what does a cell do with these proteins? So here is a neuronal cell, and this cell was incubated with a solution containing monomers of this protein called amyloid beta, this Alzheimer's protein. So let's see what the cell does. The same solution that formed these beautiful fibrils you saw earlier, right? What does the cell do with this? So here's the cell. It's taken up the solution. And this is a wide field microscopy image, so let's zoom into this part. And so here's the image, and you see not very much. But with this technique again, with direct stochastic optical reconstruction microscopy, which is this single molecule trick, we turn this picture into that. And you see that inside the cell it's totally different. So you have some fibrils that have still formed inside, but mostly the cell seems to try to deal, right, with the protein and prevent it from aggregating. So already you see it's vastly different information you get. And so the question is, what is the cell doing? Why is it doing that? I won't talk more about this because I'm coming back to it at a later stage. What I want to say is, I had to take 10,000 images to generate this one image. And nothing must move in that time. So what I had to do is to fix the cell, which means the cell is dead when I'm imaging it. It takes about 2 or 3 minutes with good equipment to take one of these images, so nothing must move. So it's not a great technique. It's very, very high resolution, this technique. So we can get 10 nanometer resolution, which is excellent. It's the highest resolution of the super resolution methods, but it's slow, right? That's one thing to keep in mind, because we have to take lots and lots of images of all the molecules until we've imaged them all. So keep that in mind. I just want to show you another example of work we've done. So this is another process where protein assemble. into something that you don't really like to have in your body, which is a virus particle. So a virus particle contains proteins that form these highly regular structures. There's a capsid structure in the middle that forms these beautiful geometric structures which contain the DNA of the virus, which is then injected into the host cell, and then and so it is surrounded by a whole layer of proteins. And so if you take an electron microscopy image of of this virus particle, here's what you see, right? So you get nice ultrastructure. You see the shape, you see the capsid in the middle. But what is hard to do with electron microscopy is to be specific, to say which protein is where in here, right? That is a hard thing to do with electron microscopy, because labeling specifically is a hard thing to do, and getting all the proteins, but that's not a problem for optical techniques, right? So high resolution, but low specificity and sensitivity for electron microscopy. Light microscopy now has this problem. It is high specificity and sensitivity, but low resolution, right? So this is a standard image. So on my camera, you would see something like this, a highly pixelated image, because these virus particles are tiny, 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 right? So these are the sort of images you would get. But again, if I have a bunch of fluorophores sitting here, a bunch of fluorophores sitting there, and so on, so probably there's a virus particle here, probably there's one there, but I can't say much more. But instead now, I localize, and you turn this image into this. So you can now clearly see the virus particles. I'm still not as good as electron microscopy, obviously not, right? But much better than before. So we can see this is a particular protein in the tegument layer, in the envelope layer of this protein, and of course they are all very regular structure. This is a virus called HSV1, herpes simplex virus 1, because it's cold sores. And most of us have it in our bodies. It's 80% of the population has this latently in their bodies, and sometimes it breaks out when you get ill. So I wanted to show you one trick that you can do with this single molecule localization microscopy trick, which is regularly done in electron microscopy. Often in electron microscopy, what you do, if you want to push the resolution and sensitivity higher, if you have lots of very similar particles, you average all the particles together. And then essentially you get a super resolution image of a super resolution image, because I get many, many more data points, right, that give me this thing. And this is something we've done here too. We've labeled lots of different proteins inside the virus particle with specific tags, and then imaged lots and lots of these virus particles and produced average images, and then modeled what the light data, the microscopy data that we get might look like. So if we assume that the proteins are distributed in shell-like layers, and then we put fluorophore labels on there, so we can model this by convolving these with linker sizes of the labels that we put on, so we get a 3D model of our virus that's labeled with the fluorophore. So we have a protein shell that may look something like that, and then we can say, well, if these fluorophores that are distributed like this, what single molecule localization image would they produce? So maybe it would be something like that, right? And we know that we have some localization error when we localize all these molecules, so we convolve this model with this localization error, so we get a model for our optical data that may look something like that. And by averaging lots and lots of these particles, we can say with high precision where proteins are located. So for example, if I take this protein here, this red one, and I label it at certain places, then on average, if I take thousands of particles, I will say that its radius is 121.8 nanometers. from the center of mass of the particle. This blue one here is 150.6. The green one is 153.8, and this envelope protein at 171.4. So the error bars you see are real. By taking lots of these shells, remember what I'm doing is now not saying, is this the actual shape of one particle? I'm just saying the average radius of this protein layer of thousands of particles is this radius. And I can determine this radius with a precision down to the single nanometer digit level. And that is remarkable that that's doable, right, with optical techniques now. If you compare this to the wavelengths of light, we are more than a factor of 100 better than the wavelengths of light fitting. And so from that we can get structural models which are interesting to biologists. But it's a slow technique. That's the problem. High resolution, high specificity and sensitivity, slow. We look at dead things. What if you want to look at living things and want to get higher resolution? Here's the second technique, structured illumination microscopy. This one isn't as high resolution, but it's much faster. And in many ways, this is a technique that was pioneered by a guy called Mats Gustafsson, who was also at Janelia Farm at the time. Tragically, he died. He died very young. He was an absolute optics genius, and he really should have gotten the Nobel Prize, in my opinion. This is one of the most fascinating techniques for biology at the moment. So we saw that we can think of this image resolution thing in terms of point spread function. So each point gets blurred, but we can think of this equally in terms of spatial frequencies. A point has an infinite number of spatial frequencies, but the aperture cuts these spatial frequencies. The microscope cuts some out, so only some spatial frequencies of my object are passed through the instrument. So here is a distribution. Of course, the physicists among you will recognize this as the optical transfer function of the instrument. So here I plot spatial frequencies along the x direction, along the y direction, and you can see spatial frequencies. This in the middle is DC, that's just a homogeneous, that's the DC component in my object function. And then as I go out towards the sides, I go to higher and higher spatial frequency information until I come to a cutoff frequency. And beyond that, my microscope will just not pass information. So what does that mean? So here is an image of Albert Einstein, as you know. But what really is behind this is also, I tell you that this is an image of Marilyn Monroe, right? Some of you may not know what Marilyn Monroe looks like. You're not old enough. Maybe the dean knows. So, I'm sorry, but you said 1971 is when you had your first meeting here. But anyway, how can I convince you about this? So close your eyes almost completely and squint at this image. Just close them completely so you see nothing, and then open them so you just see a little tiny bit of light coming through. You really have to close them almost completely, and then look at this image, and you will see, once you blur or take your glasses off if you have them, squint completely. I mean, really, you have to get a completely blurred image from this. And you will see Marilyn Monroe. Now, why is that the case? Can anybody explain that to me? Einstein has lots of hair like this. High spatial frequencies. The information changes very quickly in space. Marilyn Monroe has a very smooth skin and smooth hair, right? So she's encoded with low spatial frequencies. The frequency content in Marilyn Monroe is much lower, the spatial frequency content, than in Einstein. So the same effect you see if I reduce the image size, you'll see that, you know, from a certain distance, this looks like much more like Marilyn Monroe now, maybe a little pixelated, right, than before. Same thing happens in the microscope. Right? It's an imperfect system, and some of the information doesn't go through the microscope anymore. So how can we recover this information? Well, I have to shift some of that high frequency information into lower frequency information and then pass it through the microscope. Is that doable? Very easily. Think about this point here. If I have an object that contains wiggly lines, this is this lower structure here, right? Wiggly lines. There's a high spatial frequency in this direction and a low spatial frequency in this direction, in this image. And now, rather than illuminating this image with a homogeneous light field, let me illuminate it with a patterned light field. So now I have a spatial frequency pattern in the illumination, and I project it onto my sample. So now I get a product. I get the product of the excitation light with the object function, right? And so this gives me now, in the Fourier plane, gives me sum and difference frequencies of the spatial patterns. And the difference frequencies, you can see here, they're very low frequencies. They're called moire patterns, right? These patterns. You see them sometimes if in gardines, if you have gardines that fold over, you don't see the small squares in them, because they're too fine. The mesh is very fine, but once they fold over, the meshes beat. The frequencies of these meshes beat, and you can see these low frequency patterns. And of course, if I know the spatial frequency for my illumination pattern, then it is clear that I should somehow be able to recover from these low frequency beats the original frequencies in my sample. So you have to play some Fourier tricks. It becomes a mathematical problem then rather than an optical problem. And so you move these illumination patterns quickly along to generate lots of images, right? You have to rotate them as well to be able to do this. So here's this. I haven't got really time to do this. So an image is really the object times the excitation light in real space. That's what we just seen. So in Fourier space, this becomes a convolution again. So the frequency component in the excitation field are convolved with the frequency components in the object field. So you see the idea here. If I have a sinusoidal pattern and an object, so this object may have spatial frequencies like that. This is the spectrum of the object. A sine wave, right? An intensity that is modulated sinusoidally has a DC component and the frequency components, plus omega and minus omega, right? So 3 delta functions. What's a convolution of that function with this? What's a convolution of a function with a delta function? It's just a function centered on the delta function. So we'll take that function, and now there will be a copy centered on that delta function, one copy centered on this delta function, one copy centered on that delta function. So now what I get is I get a mess, right? I get this, the original function is here, the original function is over the center one, and the original function is over there. Why would I want to do that? Well, I tell you why. Because the microscope, I shouldn't use that word. So the microscope cuts off frequency information beyond the cutoff frequency here, right? That's how many frequencies are passed. So this will pass Marilyn Monroe here, but if you want Einstein, you need these tails here too, right? These high frequency tails. They're gone in the original microscope. But by doing this trick, you can see I'm shifting this function over here, and some high frequency information is now shifted into the passband. And this function here is shifted to that side, so this high frequency tail there is now also shifted into the passband. So now the trick becomes just to be able to untease that information, and that requires lots of calculations. So it's a computationally very intensive method, but in practice it's very simple. We use spatial light modulators and sort of a little bit of optics, right? And we very quickly project patterns, lots of patterns onto our sample at the highest spatial frequencies we can do with our system. The finer the patterns, the better resolution, right? And we rotate the patterns into different directions and then get the information back. And that then allows us to look at things in time, because I need 9 images to generate one super resolution image. And with a good camera, a good system, I can take up to 150 images per second, right? With a scientific CMOS camera, that's about as far as I can go. And so if I roughly divide that by 10, I can go up to, if you do it really, 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 really well, I can go up to 15 hertz with this technique, right? Here's a slow example first, but it's an interesting one. So here again, we have these amyloids, and one interesting question in these amyloids is if you have a certain fibril, does it cause other proteins to misfold and grow these fibrils? And you can see this here. In red, we have fibrils of amyloid beta, and in green, we have a monomeric solution of protein that's healthy. But the presence of this fibril forces this healthy protein to adopt the wrong shape and then grow these amyloid fibrils in time. And this you see here. So what you're seeing here are molecules growing in real time. So you have the original seed species, and in green now you see how the amyloids are extending in time. So the molecules come from the solution, attach, and grow. So you look at molecules, and once you see stuff like this, you see interesting things. For example, I leave this up to you to decide. I don't have time now because I need to finish. But you can see actually with these imaging techniques. So if these are time, this is a time axis down this way, and this is distance. And you can see these fibers actually grow much faster from one side than from the other side. Why is that? It's interesting questions. Amyloid beta does that, other amyloids don't do that, right? So this is polarized growth. You'd say, well, but this is still boring though. Look at these time scales, this is really slow, right? So what do you bother with this structural illumination trick? You just told us it's so quick, but in fact you could probably just do, this is not really a test for the technique, and that's true. So the funny thing is, this is what happens thermodynamically, right? This is a kinetic process. This is a kinetic process that's just physical chemistry. You have a solution of the stuff, you put it on the cover slip, and it grows. What happens inside a cell? So let's look at that cell that's taking up these amyloid fibrils. And now we use structured illumination. We can see the individual fibrils inside the cell. These are amyloid beta fibrils. Just that protein, none of the other proteins, none of the other 10,000 proteins inside the cells are touched. It's just that A beta protein. And now look at this time scale. These are seconds. So we're taking here, this is at 12 hertz, 12 images per second super resolved. And you can see that the cell is screaming, right? This cell here, this one cell, here's the nucleus. It's really trying to deal with these invaders somehow and doing something. This is of course stuff you would never see with any other technique, with the conventional electron microscopy or even storm. And so you see actually interesting things. The cell tries to push these particles towards what's called the microtubular organizing center, presumably to target them for destruction. So there, the molecules get labeled with a protein called ubiquitin, and then the cell knows that those things should be degraded. The problem is that these amyloids are extremely strong, and the cell can't dissolve them. So we wanted to ask more questions about this. So we see this interferes with nuclear division and so on. But we found then that every amyloid gets packaged into what's called the lysosomes. The lysosomes are vesicles that contain lots of proteins inside and enzymes to break down products inside the cell that the cell doesn't want, right? And so the amyloids are clearly one of those things. So we have labeled the lysozymes here in green with a protein called LAMP1. So LAMP1 is the lysozyme associated protein here with a GFP, with a green fluorescent protein. So they appear as these green dots. So each one of those are these vesicles containing stuff. And the amyloid beta here is in blue. And so if I look at this, these are SIM images, structured illumination images. We can see that wherever I have an amyloid, there also seem to be vesicles, right? There seem to be these lysosomes. So that all of these amyloids here seem to be packaged inside lysosomes, right? And then you turn on the camera. This is 2 color now at very high speed. So you see again, this is seconds. So you see these lysosomes trafficking. You probably can see that there are 2 colors in this spot here, or in that one there. They're whizzing up and down inside the cell. So each one of these lysosomes you can now watch as it's moving up and down inside the cell. And the cell's trying to deal with it. And you can then image. So inside cells you have what's called microtubuli. These microtubuli are essentially the motorways along which all the trafficking takes place. So we've imaged in live cells here that you see with a protein called LifeAct. We can see directly the microtubuli. You can see it's a network that very rapidly reorganizes. All these microtubuli are moving around. They're polymerizing, depolymerizing at ridiculous speeds, right? So it's a highly dynamic process. This is real time. And you can see the particles moving up and down, these A beta particles inside. And if I move in and zoom in, so you can actually see, you can see how they track and trace these individual, they go along these highways. So these are things that, you know, just about 2 or 3 years ago, you couldn't have seen with other techniques. So there are very interesting questions about this, right? Because it seems that all these lysosomes are then accumulated at certain domains inside the cell. Then the whole cluster of aggregates gets packaged with the lysosomal membrane. And then actually you get problems because the cell can't deal with it anymore. So I want to stop by just saying the few key points. So the first part of the talk showed you that you can gain super resolution by imaging individual point spread functions and imaging them sequentially, trading speed for resolution. With that technique called single molecule localization microscopy, you get 10 nanometer resolution. 10 nanometers, so that's very high. And if you average data, you can get even higher localization precision. Structural illumination produces spatial frequency beats. And these new spatial frequencies encode information. So from higher frequencies that are not passed by your microscope in lower frequencies, which are passed by the microscope. And then you use Fourier methods to tease out the high frequency information. And all of these techniques you trade. So with this technique, you can get about 100 nanometer resolution with the structural illumination method. But it's very, very fast, up to 10 hertz. But you can see from those two, super resolution is always gained, no matter which technique, by trading off temporal with spatial resolution. And then finally, I mean, I hope I could convince you that these techniques are really offering completely new insights into the way living systems work at the molecular level. And here are the guys. This is Eric Betzig. This is Bill Murner, who won the Nobel Prize for the single molecule localization trick. This is Stefan Hell for another, who won it for STED, for stimulated emission depletion microscopy, which is another superresolution method. And this is Mats Gustafsson, who I think passed away 2 years ago and was far too young. And with that, I stop and take questions. Thanks.
Thank you very much, Professor, for this excellent, fascinating talk. And we have time for a couple of questions.
What do you think is a potential for each of these techniques to go deeply into the tissue, for example, or into the sample?
It's a very good question. So in the first technique, it is your ability to get accurate images from the point spread function. So if you distort your point spread function because you go through lots of tissue, then you cannot accurately fit the point spread functions anymore. But so if you can go down to 10, 15 microns with the first technique. In some instances, if you have benign samples, you may go a little bit deeper. But then you start distorting the point spread functions, and you cannot get decent data back. because you need to be sure, right, that the pattern I see is associated with the molecule, so you have a model for your point spread function. If you can't fit that model anymore, then you're done. The second technique has also that problem. You need to be able to get the patterns faithfully reproduced. But there's another issue. The way we produce the pattern is by interference of two light waves. And the way we do this, we have two laser beams, and we place them right at the end of the back focal plane of the illumination objective. And then they come through, and then they meet in the sample. You know, they go through the objective, meet in the sample, and interfere and form a standing wave. And the best resolution we get if you pull these points as far apart as possible. And what will that be? That just gives you a frequency of lambda over 2Na. Lambda over 2Na, right? So, and you get that when you actually just use the full Na of the objective. That's actually what limits the resolution to about lambda over 2 in the, well, it gives you a factor of 2 over the things. So, and the problem with that is that, so you go very close to actually to the conditions where you are in turf mode with high objectives, and so the patterns will only be produced at a very shallow depth. So it'll work, you get good contrast for your patterns of about, up to about 5 to 7 microns. That's not great. But then you can combine these ideas of structural illumination with other techniques such as light sheet imaging. And with that, so for example, Eric Betziger has done that with a technique called lattice light sheet imaging, which was a science paper a year ago or so, where you can go deeper with this principle, but not quite so high resolution.
Do you have some more questions?
I have one. So is the problem to localize your points in the first technique? Like, do you have to cool the cell down a lot? Like, how does it work? Because even a tiny motion is going to spoil the image a lot.
Absolutely. So that's why you fix the cells. So you can really not have anything moving during the time you localize. But typically, so the way, I haven't told you how you switch molecules on and off. The first technique relies on you being able to switch the molecules off and just switching a subset on, right? So what you do is you chemically reduce the fluorophores. They go into a dark state, and then I use another laser to activate just a few fluorophores. And so when one fluorophore is active, I image it for as long as it's bright, so typically 1,000 photons or something I might get from a biological fluorophore. And those you take, typically that emission pattern will be over 10 milliseconds. Over 10 milliseconds is what the sample needs to be stable for each fluorophore, right? That's usually not a problem. And actually, there are finer points, but actually the patterns are massive, right? So even if you have motion around, you know, it's the center that doesn't need that mustn't change. That's the most important thing. Yeah.
Is it possible to apply the first of your techniques in order to enhance the resolution of the scanning electron microscopy?
Ha! Now you're asking.
Because you can illuminate, I think, one point at a time.
Well, I mean, there's something fundamentally different in the way the image is generated in the scanning electron microscope. But in fact, it's not such a silly thing to think about similar tricks in other domains, right? In actually in astronomy, people have used this trick for a long time, right? To differentiate two stars, right? So as long as you have, all you're doing is you're localizing patterns in a better way. So whether that would work in scanning electron microscopy or not, I wouldn't be qualified to tell you right now.
Some more questions?
So what is the selection of technique for illuminating only single molecule? So what is the requirement for the illumination technique? How you do it?
Actually, it's remarkably simple. So that's why the first technique is so popular, because it's actually easy to implement. So what you do is just a standard microscope, right? And you use a laser usually to put a lot of light on. But The trick is in the photochemistry. So you switch just molecules off, and then so you need a photoactivatable dye, such as Alexa 647, for example. Cyanine or oxyzyme dyes work, the rhodamines work in some cases. And then you put a thiol-containing compound, a reducing agent such as MEA, mercaptoethylamine, onto the sample. And so once you excite the molecule first, you go from a singlet to a singlet state, right? With normal light, normal excitation. So you get a flash of light, but after a couple of cycles, you know as a physicist, right, it will go from the singlet to a triplet state. And in that triplet state, it reacts with this thiol-containing compound, and it forms a bond in this excited state, and it immediately turns dark in that state, as long as the thiol is attacking. And then you use just a different color to kick off that thiol again. So first you get a flash of light, the molecules cycle, you reduce them down, and then they're dark, until all molecules have done that. So first in the standard microscope, you get a flash of light, then everything is dark, right? And then some come back into the singlet state, and then you can just image those few that come back. And you can reactivate another subset with a different color, kick off the thiol bond, and so on. So all you need is usually you have a second, what's called an activation laser, and you have a normal excitation laser, just a normal microscope. Sometimes you don't even need to operate the excitation laser because there's a bit of oxygen left inside the sample and reoxidizes the system. And so then it becomes active again.
So spatial selectivity is not needed for illumination?
No. You don't, of course, know which molecule will come on next. It's stochastic, right?
Yes.
But it doesn't matter. All you need to do is control the rate such that the likelihood of two proximate molecules coming on is low. So that gives you the light switch to do that, essentially.